I see. Well, it sounds still like there's a lot of there's a lot of stuff that goes on from the time that you guys get that raw data and you sort of do your quality control, your QC check, before I see it on my 23andMe profile page. I mean, is all of that just sort of computational biology at that point? Who's looking at that data? Who's organizing it? What's sort of going on there?
Yeah, it really becomes a computational biology problem once we get the data from the lab, and that's really where 23andMe concentrates. Most of our staff here are computational biologists or software engineers, so that's really where we're best. So the next major component there is called calling. That's turning the data that we get from the lab into the AAs, AGs, or GGs that you can download in your raw data or produce the reports you see on our website. And that's kind of an involved process, but it, in essence, relies on looking at these XY plots and making sure that everything is where we expect it to be.
And so the final results that we get back seem to look so curated and so official. I mean, how much of the computational sort of side of this is sort of deriving, oh, what does this letter mean? I mean, are you guys reviewing all the literature and selecting things, and how are those decisions made in general?
So we actually have a pretty big content team at 23andMe. They're the ones who scour the literature, look at what's going on in the current science of genetics, and find out, oh, this mutation or this variant causes this disease and it's a new discovery. Then they would write up what they think about that, try to put it in language that makes sense to our users, and if it meets our scientific criteria, it goes out to the website. It is all automated. It can take time just to process the data through to the website, but the actual process of taking your data, calling all of the variants and all of that is quite quick. You can think of the website as being in two parts. There's the part where we just look at the calls that you have for disease, and the report just links that call to the result. So for example, we could say, you are a carrier of cystic fibrosis because you have this variant. So that's a scientific challenge to figure out what's going on in the literature, but it's not an algorithmic challenge. The other part of the website includes things like our ancestry composition feature and our relative finder feature. Those are difficult algorithmic problems. You want to take the whole genotyped sample, all million variants, and try and figure out, based on that, what is this person's ancestral composition? So you can imagine that that's actually a more complex task and requires a lot of code taking it from A, B, C through to the end.